Good evening, everyone. So, this week we are continuing our lecture on basement membrane zone. Last week we discussed the formation of desmosomes. Now, desmosomes are adherents molecules which attach two different adjacent cells together. So, imagine if a cell is like a room, the desmosomes attach one wall to another. This week we'll learn about hemidesmosomes. Hemidesmosomes attach the floor of the cell to the underlying proteins and molecules and further strengthens the attachment of the cell to the basal layer and further to the basement membrane. Okay. Now, all uh, of all the proteins that constitutes the hemidesmosomes, if there's any problem in those proteins, let's say you are a patient has an acquired mutation in one of those proteins, the basal cell layer will not be properly attached to the floor and it will get separated from the underlying dermis. So, in all the acquired blistering disorders that have mutation in the hemidesmosomal proteins, the split is in between the epidermis and the dermis, or a subepidermal split. Okay. If there are any autoantibodies against these molecules which form the hemidesmosomes, the split again will be between between the epidermis and the dermis, and you will have a subepidermal split. Okay, so that is why in disorders like bullous pemphigoid, mucous membrane pemphigoid, cicatricial pemphigoids, you have the split in between the epidermis and the dermis because there is problem in the proteins which constitute the hemidesmosomes and the proteins that traverse or cross the lamina lucida section of the basement membrane zone. So today we are going to learn about them. And when we learn about them, we can now correlate why we see a subepidermal split, why we see the different kind of clinical features like a tense bulla in bullous pemphigoid or a fluid-filled, not ruptured bulla, as opposed to ruptured bulla in pemphigus. Because in the last week video, we discussed that desmosomes are the structures which connect two cells together, two adjacent cells together. So when there's a problem in desmosomes, the split will be either intraepidermal or just above the suprabasal layer. Okay, and we also discussed how the row of tombstone appears in a biopsy because of this desmosomal problem. But in hemidesmosomal problem, you will have a split between the epidermis and the dermis. And for us to understand the clinical features of subepidermal blistering disorders or acquired subepidermal blistering disorders, we need to know how a hemidesmosome is formed and how do the basal cell of the epidermis attach themselves to the dermis. Okay, so let's, without much further ado, we'll start our discussion on basement membrane zone part 2, that is hemidesmosomes and the lamina lucida proteins. So as I've already said that desmosomes are intercellular adhesions. That means the attachment will be between two adjacent cells. The hemidesmosomes are responsible for attachment of basal cell lower membrane to lamina lucida complex. Okay, so what essentially happens is if these are your basal cells, desmosomes will be here and hemidesmosomes would be here at the basal level, at the floor of the cell and attached to the further complex of proteins in the lamina lucida. Okay, so that is the function of hemidesmosomes, to attach the floor of the cell to the underlying tissues, underlying proteins. They're known as hemidesmosomes because initially it was thought that they look like half of the desmosome. So if we magnify this area, it will look like this, a bit like this. And if we magnify the hemidesmosomal area, it will look like this. So it looks essentially like half part of desmosome. That is why the name hemidesmosomes. So this is an electron microscopy graph or electromicrograph of hemidesmosomes, and here you can see these darker hump-like regions, which are the hemidesmosomes. The lighter shade layer is the lamina lucida, or the translucent layer. Okay, the lamina lucida, and and in this video we'll keep our discussion limited to lamina lucida. We'll discuss the lamina densa and the anchoring fibrils in the next last part of this series, which will be there in next week. So all of these hump-like areas are actually hemidesmosomes attaching the basal layer of the cell to the underlying group of proteins. Now, one doubt was raised in last week. What's the difference between anchoring filaments and anchoring fibrils? Remember that anchoring fibrils are larger, they are loop-like. Okay, they are loop-like, and they are present in the papillary dermis or the reticular dermis. They are essentially a part of dermis. Anchoring filaments are the proteins that cross the lamina lucida. So here are the anchoring filaments. Clear? Let me just remove these markings so you are able to see the anchoring filaments properly, and also anchoring fibrils. So this is the difference between anchoring fibrils and anchoring filaments. Fibrils are in the dermis. Filaments cross the lamina lucida. They are above. So this is again the same picture that we discussed last week. There are four zones to basement membrane. First is basal keratinocytes, which essentially house the desmosomes. The lamina lucida, which house the hemidesmosomes. Hemidesmosomes are also present here also. It's a connection between basal keratinocytes to lamina lucida group of proteins. The third is lamina densa, which is the blue one. And then fourth are the anchoring filaments, which are present in the level of dermis. Okay. So this is essentially a recap from last week's video. So, in basement membrane zone, we have the four zones: keratinocytes, lucida, densa, and sublamina densa, or the area where we have the anchoring fibrils. And these are the major constituents. We have discussed a little bit about keratins 5 and 14. We have discussed how they lead to formation of desmosomes at the lateral wall. We will restrict our discussion this week to the laminar lucida and hemidesmosomes, and these are the proteins: BPAG1, bullous pemphigoid antigen 1, Plectin, bullous pemphigoid antigen 2, integrins, and laminin 332 to some extent. We'll touch laminin 332. So this will form our discussion for this week. This is a diagram that I've taken from Bologna, if I remember correctly, and this is essentially the basement membrane zone. So initially, we have discussed about the keratin filaments. These are your keratin 5 and 14 filaments, and they attach and this structure is the hemidesmosome. Okay, you understand now how it is at the basal level. So in hemidesmosomes, the first connection is between the keratins and the plectin plate. This is the plectin plate. It's formed by a lot of plectin molecules, which has a globular head and a long filamentous tail. Okay, this is a plectin plate. Another protein here is bullous pemphigoid antigen 1. You can see it here. We'll discuss each of them in detail, don't worry, in subsequent slides. So the keratin filaments are intertwined in the plectin and this form the inner hemidesmosomal membrane. The outer hemidesmosomal membrane is here, outer hemidesmosomal membrane, and that is formed by CD151, the heads of the alpha 6 beta 4 integrins, and also collagen 17, which is also known as BPAG2, bullous pemphigoid antigen 2. Only the head part. So this is the outer membrane, outer hemidesmosomal membrane, and the rest part is lamina lucida. So we'll discuss lamina lucida and the hemidesmosomes in this video. So let's start our discussion by discussing in detail the structure of hemidesmosomes, and I've tried my level best to make it simpler so that it is easier to remember and you can easily reproduce this in your exams with an easy to understand diagram. So let's start with hemidesmosomes. Now hemidesmosomes, the two major proteins in the inner hemidesmosomal membrane is bullous pemphigoid 230, also known as bullous pemphigoid antigen 1, and plectin. Now 230 is the molecular weight in kilodaltons. So, BP230 is the heavier protein as compared to BPAG2, and it is also known as BPAG1. They essentially form the same. They function the same as Plectins in desmosomes. So what Plectins used to do is to attach desmocolin desmoglin to desmoplakin. So this is what they are going to do. Bullous pemphigoids are going to attach the keratin filament proteins to the underlying to the underlying integrins. So that is the function is essentially the same as Plectins in desmosomes. So BP230 bind to keratin filaments which are present inside the cell. So this is present inside the cell. The plectin also binds to keratin filaments, and plectin plus BP230 forms the inner hemidesmosomal membrane, the plate which is inside the cell, and attaches the keratin filaments. Clear? Let's move forward. So plectin are the major protein of inner hemidesmosomal plaque, and mutations in plectin causes epidermolysis bullosa simplex, EB simplex. But two essential features are added. So in order to remember it properly, P for plectin and P for progressive muscular atrophy, P for pyloric atresia. So now you'll be easily able to remember that if there's a mutation in if there's an acquired mutation in plectin, then you will have EB simplex. Remember if you have read about little bit about epidermolysis bullosa, you will understand that simplex is a bit superficial split. Junctional is at the junction of the epidermis and the dermis, and dystrophic is somewhat, it's a deeper split. So the first split to come in EB simplex would be a bit superficial, and superficial splits are simplex. So epidermolysis bullosa simplex. And if the mutation is in plectin protein, then it is associated with progressive muscular atrophy and pyloric atresia. So remember, P for plectin, P for progressive muscular atrophy, and P for pyloric atresia. Just remember this. Clear? Let's move forward. Bullous pemphigoid antigen 1, also known as bullous pemphigoid 230 kilodalton protein, is also a major protein of inner hemidesmosomal plaque. So when I say it's major protein, remember that inner hemidesmosomal plaque can have a lot of different proteins, but we will restrict the discussion to BPAG1 and plectin because these are very important for hemidesmosomes, essentially most important. So we'll discuss only these two proteins in the inner hemidesmosomal plaque. The globular end. Remember that all the proteins have somewhat of a big globular head and a long filamentous tail. Okay, most of the proteins in this structure. So in BPAG1, the globular end binds to keratin filament, and the filamentous end has a centrally coiled structure. So this will be coiled. Okay, the amino terminus, that means the further end of BPAG1, binds to the cytoplasmic domain of BPAG2 and integrin B4 and urbin. Urbin is required for ERB2 signal. You just have to know this. You need not remember urbin. So what essentially happens is you have a cell, and at the basal level you have the inner hemidesmosomal plaque that has plectin and BP230. Okay, this will attach to BP180, which is the bullous pemphigoid antigen 2, and integrins. So these are integrins, and this is BPAG2. Now BPAG2 is a transmembranal protein. That means it will cross the membrane. So BP230 serves as an attachment of integrins. These are integrins. These are another molecule of the hemidesmosomal plaque, and this is BPAG2. So this is where the attachment essentially occurs. Okay. The neuronal isoform. So there are various forms of bullous pemphigoid antigen, and there's a neuronal isoform that has been present in neurons, the attachment of interneuron attachment. So that neuronal isoform is referred to as BPAG1N or dystonin. So that is why in disorders like bullous pemphigoid, when you have antibodies against the bullous pemphigoid antigen, some patients can present with neurological problems. Why? Because the antibodies are also targeting this neuronal type, the dystonin type, and the problems with brain also start happening along with blistering disorders, because another form of bullous pemphigoid antigen is present in the brain, BPAG1N. Okay, mutations in this protein causes epidermolysis bullosa simplex. Clear? Because it is very superficial protein, so the split will be superficial, so the disorder will be EB simplex. Now, we have discussed inner hemidesmosomal plaque. Now we'll go forward with the outer hemidesmosomal plaque. Okay. Now remember that the outer hemidesmosomal plaque is not outside the cell. It is known as outer because it is closer to the outside. So the plaque which is inner or closer to the inside is inner hemidesmosomal plaque, and the plaque which is outside, closer to the outside but not outside the cell, is outer hemidesmosomal plaque. Okay. Now bullous pemphigoid antigen 2 is like a hockey stick. It has a head and a tail. Okay. So the globular intracytoplasmic head, that means the globular head which is inside the cell, it binds to integrin beta 4 and plectin. So it binds to plectin, it binds to bullous pemphigoid antigen 1 and also integrins. Okay. The filamentous or the long end, a central rod with a flexible tail, is a collagenous extracellular domain which binds to laminin 332. Now laminin 332 is present in lamina densa, laminin, lamina. Clear? So laminin 332 is present in lamina densa, but a part of it is protruding towards the cell and attaches to the bullous pemphigoid antigen. Remember? So, what essentially I'm trying to say is that plectins and BPAG1 forms a complex. The complex attaches itself to bullous pemphigoid antigen 2, and this antigen 2 also attaches the integrins in the side. And since bullous pemphigoid antigen 2 is a hockey-shaped molecule, it will go downwards and attach to the laminin 332 present in lamina densa. Clear? We will show you an image and it will be easier to understand. The rod domain inserts into the lamina densa and then turns back through the lamina densa into lamina lucida. So what essentially happens is you have a cell and you have the dense lamina densa. The middle part is known as lamina lucida. So bullous pemphigoid antigen will go back and then loop back like this, like a hook. So it hooks a line. Okay. Autoantibodies against bullous pemphigoid antigen 2 causes bullous pemphigoid, okay, pemphigus gestationis, linear IgA bullous disease, and here the antibodies are against the N-terminal 16A domain. Okay, if the mutation is in distal carboxy terminus, it causes mucous membrane pemphigoid. Now, mucous membrane pemphigoid has shown autoantibodies against two parts, the NC16A domain, the same autoantibodies that we see in bullous pemphigoid, PG and LABD, and additional antibodies to distal carboxy terminus has also been found in mucous membrane pemphigoid. So these two are the examples of autoantibodies to bullous pemphigoid antigen 2. Clear? Let's move forward. So bullous pemphigoid antigen 2 is also known as transmembrane collagen. Why transmembrane? Because it crosses the cellular membrane. Okay, so you have the basal layer and then you have the BPAG2. So since it crosses the cell membrane, it is known as transmembrane. Since this is collagen 17, here you can have the transmembrane collagen 17. So this is the collagenous domain, also known as collagen 17, and it is going outside, crossing the membrane, so transmembrane collagen. The collagenous domain is extracellular, that means it is outside the cell. And bullous pemphigoid antigen 2 is responsible for regulating interaction between the basal cell and the basal membrane, hair follicles, and the melanocyte stem cells. Now you can see in this rough schematic diagram that bullous pemphigoid antigen 2 crosses the cell and goes downwards towards the basement membrane. So it's an important molecule to regulate this interaction. We are clear on that? Let's move forward. So inside the cell, the bullous pemphigoid antigen 2 or BP180 will bind to plectins plus BP230 plus integrin B4, beta 4. Outside the cell is going to bind to the integrin alpha 6 and laminin 332. So what essentially happens is you have the inner hemidesmosomal plaque. Let me change the color for better understanding. And you have the BP180 and this is going to loop. Okay. So it will attach to bullous pemphigoid antigen 1, plectin, and the beta subgroup of integrin. Outside the cell is going to attach to the alpha subgroup of alpha subgroup of integrin and the laminin which is present in the lamina densa. So this is essentially the structure of a hemidesmosome. It's a major antigen for bullous pemphigoid. If there are autoantibodies against this particular protein, you will have bullous pemphigoid. And if there's a mutation in this particular protein, you will have junctional epidermolysis bullosa. Now remember, bullous pemphigoid antigen 1 mutation causes EB simplex. BPAG2 mutation causes junctional EB. Why? Because it is now crossing and reaching till the lamina densa. So the split will be thicker. It will be severe. It's at the junction of the cell and the dermis. Remember, just below lamina densa, you will have dermis. So the split is acting at the junction. The split is between the lamina lucida and the densa. So you will have junctional epidermolysis bullosa. Okay, let's move forward. So let's come to integrins. So we have discussed BPAG1, BPAG2, plectins. We'll discuss integrins now. Now integrins are protein molecules. They are formed as a dimer, a two type. One is beta, one is alpha. The most important integrin in hemidesmosome is alpha 6 beta 4. So that is the sixth isoform of alpha and the fourth isoform of beta. Clear? Now, integrins are essential for integration of hemidesmosome to the underlying dermis. That is why the name integrin. It is important for integration of hemidesmosomes to the underlying dermis. Okay, it integrates the hemidesmosomes and the basal cell to the underlying dermis. So they are known as integrin. And since there are two subunits, alpha and beta, integrin alpha 6, beta 4. The beta 4 subunit is responsible for attachment to plectin and bullous pemphigoid antigen 2. Okay, so again, this is plectin, this is BPAG1, this is BPAG2, and here you will have the alpha unit. And we have already explained to you that alpha unit crosses the membrane and attaches to bullous pemphigoid antigen 2 also. Clear? The alpha, sorry, the subunit alpha 6 isoform A are secreted by keratinocytes. So this protein molecule is formed by keratinocytes and then let out of the cell inside the lamina lucida. It binds to the NC16A region of bullous pemphigoid antigen 2. Clear? The alpha and beta subunit form pockets for ligand binding. So alpha and this one is then beta, and they will form a pocket so that other ligands can bind at this pocket. The integrins show very high affinity to laminins 111 and 332. They are the preferred ligands. So at one end, integrins are attaching themselves to plectins and bullous pemphigoid antigen 2, which are part of the hemidesmosomes. At the other end, they are attaching themselves to laminin, which is part of the lamina densa. So they are very important molecules to attach the basal cell to lamina densa. Clear? So mutation causes junctional epidermolysis bullosa with pyloric atresia. Now remember that integrins attach to plectins and the plectin mutation causes EB simplex with pyloric atresia. But if there's mutation in the integrin, then it will cause junctional EB with pyloric atresia. Okay, remember it will be easier to understand because integrins are the molecules situated below plectins, so the split will be distal and it will be junctional EB. Clear? So anything which is happening at the level of lamina lucida just outside the basal membrane will lead to junctional epidermolysis bullosa. Clear? So we have essentially discussed, we have gone from the cell hemidesmosome outside the cell lamina lucida. We have just reached lamina densa, but we'll discuss one important protein by the name of CD151, also known as tetraspanin. Why tetraspanin? It essentially has four moieties. It's like a structure is like this, and it spans everywhere. So four arms and spans everywhere, and it's responsible for connecting different protein molecules of the hemidesmosomes. So it's a widely expressed cell surface transmembrane proteins, belongs to tetraspanin family. It forms complexes with various integrins. For example, integrin alpha 3 beta 1 and integrin alpha 6 beta 4. Now let's say this is the cell, the basal layer of the cell. Okay, and you have integrin alpha beta, and this one is alpha 6 beta 4, and this is the hemidesmosomal plaque. Clear? There is another integrin by the name of alpha 3 beta 1, which is not a part of hemidesmosome. It is entirely separate and it is away from hemidesmosomes. Okay, and CD51 will attach to both of them. Let me change the color. CD51 will attach to both of them and provide further stability to the attachment of the cell to the underlying tissues. If it attaches to the hemidesmosome, then the hemidesmosome will grow stronger. If it attaches to integrin alpha 3 beta 1 without the hemidesmosome, then also it will act as further points of attachment. Okay. It has been found in various studies, and I think this is very interesting, that in animal studies in which CD151 was neutralized or removed, okay, so it was found that the laminins, which normally attach to integrin alpha 6 beta 4, they started getting attached to alpha 3 beta 1 or the other junctions. So this is the other junction. Let me just show it to you here. This is the other junction. When CD151 was reintroduced into the animal model, the attachment of laminins shifted from alpha 3 beta 1 to alpha 6 beta 4 and the hemidesmosomes were formed. So it was postulated that CD151 is a very important molecule for formation of hemidesmosomes. Okay, since it has four large arms to collect all the proteins, it will form hemidesmosomes. Okay, and it is a very essential protein for the structure and the assembly of hemidesmosomes. Am I clear on that? So that is the importance of CD151 or tetraspanin. It is important for assembly of basement membrane, kidney, skin, inner ear. It is very important for erythropoiesis also. The main function is to provide a framework for spatial organization of different hemidesmosome components. So to provide a framework, and on top of that framework, other proteins will get together and attach and will form a good hemidesmosomes. Okay, so that's why I was telling you about the story in different articles where CD151 was removed and you had no hemidesmosomes, only attachment via integrin alpha 3 beta 1. But when you introduce CD151, the attachment shifted to integrin alpha 6 beta 4 to form hemidesmosomes. So if you want to have hemidesmosomes, CD151 should be there. So we have gone from the base of the cell, we have gone from the integrins and BPAG2, which goes from the inside the cell to outside the cell. Now integrins and BPAG2 will attach to laminin 332 and this is present in lamina densa. Since it's a part of lamina densa, it will be discussed in next week's lecture. We'll just touch upon it. So it has three chains. 3, and 2 are the numbers given to those chains. And then it's a disulfide bonded complex of chains. That is why laminin 332. Okay. These are also secreted by keratinocytes and they bind to integrins and bullous pemphigoid antigen 2. These two proteins are transmembrane. Transmembrane means they cross the basal membrane of basal keratinocytes and reach to the dermis. Okay. The mutation again causes junctional EB. Why? Because we are still at the junction of lamina, lucida, and densa. So junctional EB. If there's autoantibodies, then in 10 to 20% cases of mucous membrane pemphigoid, the antibodies were found against laminin 332. Okay, while on the major one is because of the two areas of mutation in BPAG2, 80% of the cases. In 20% of the cases, there have been autoantibodies against laminin 332. So if I want to recapitulate that if this is bullous pemphigoid 2 and this is laminin, if the break occurs anywhere here or anywhere here, the clinical feature would be essentially same. So both of the breaks causes junctional EB, while this is because of autoantibodies against laminin 332 and this is because of autoantibodies against BPAG2, but essentially the features would be same. Both of them, the antibodies, both of them will cause mucous membrane pemphigoid. In roughly 80%, the antibodies are against BPAG2, while in roughly 20%, the antibodies are against laminin 332. Clear? Laminin 332 autoantibodies have also been found in bullous pemphigoid, anti-P200 pemphigoid, and EB aquisita. Okay, so if you want to remember it, you will remember that the mutation causes junctional EB, while autoantibodies in 20% cases of mucous membrane pemphigoid have been found against laminin 332. Clear? Let's move forward. So, essentially, we have studied the structure of hemidesmosomes and how does this cell attaches itself to the underlying dermis, or at least till the lamina densa in this video. Okay. So we are going to revise how a hemidesmosome part is formed till lamina densa. Okay. So this is the intercellular space. This is the basal part. We're looking just at the lower part of a cell. Okay. So this is the basal space, and these are the keratin filaments. Okay, keratin filaments. They are very long fiber-like, wire-like, looping inside the cell, attaching themselves to the nuclear membrane, totally filling the space inside the cell. Okay, so keratin filaments. Then we have this loop structure which is known as plectin. So it has a globular head and a long filamentous tail which intertwines between the keratin filaments to attach themselves to the hemidesmosomes. Clear? Then you have the yellow one which is BPAG1. BPAG1. It has a globular head, the big oval head, and a long filamentous rod. Both of them, BPAG1 and plectin, will form a complex, and this complex will further provide other areas of attachment to different molecules. Clear? Now this attachment forms the inner hemidesmosomal membrane. Clear? We are okay? You're with me till now? Let's move forward. Now comes integrin. So this is the beta subunit, the larger one, while this is the alpha subunit, and both of them together forms the integrin. So integrin will attach to the complex of BPAG1 and plectin and will cross the membrane and reach outside the cell. Clear? Here CD151 will provide an attachment and further stability and form the hemidesmosomes. Now we have the BPAG2, BP antigen 2. BP antigen 2 will attach to integrin. It will also attach to BPAG1 and plectin complex. Okay. And then the long part will cross the cell, go till the level of lamina densa and loop back in the lamina lucida. So now you can see why integrins and BPAG2 are known as transmembrane proteins. Clear? The complex formed by the heads of integrins and the head of BPAG2, when it attaches to the complex of plectin and BPAG1, forms the outer hemidesmosomal membrane. Remember, both hemidesmosomal membranes are inside the cell. Okay, outer is because it is close to the outside, inner is because it is close to the inside. Clear? Now, BPAG2 can attach to the laminins, to the laminins directly. It need not get attached to the the plectin complexes. It can directly attach with the keratin filaments and the laminin. There are some focal points, but the major process in hemidesmosomal organization is the one in the center. So if we draw two lines, one just below, starting just below the level of the cellular membrane, till the laminin, the heads of the laminin, it will divide into three parts. The first part is hemidesmosome. So everything inside the cell is hemidesmosomes. There are proteins which extend outside the cell. They are part of hemidesmosomes, but the structure, the half semicircular or the dark humps that we showed in the initial slides, that is the hemidesmosome. So this part is hemidesmosome. Okay. The middle part is lamina lucida. Clear? And the below part is lamina densa, consisting of mainly laminins. Clear about it? Now these laminins will attach to anchoring fibrils inside the dermis, but this part, lamina densa and the anchoring fibril, will be discussed in part 3 of the video. With this we'll finish basement membrane. So now just a small 1 or 2 minutes on focal adhesions. Now what exactly are focal adhesions? So you have a basal cell and you have a hemidesmosome like this. And this hemidesmosome will attach to the laminins. Clear? There are structures, mainly Kindlin 1 and 2, vinculin, talin, which attach to actin filaments. Okay, so this purple one are actin filaments. Clear? While the blue one are keratin filaments. So keratin filaments attach to hemidesmosomes. Actin filaments attach to focal adhesions. Clear? Am I clear on that? Now these focal adhesion molecules, Kindlin, vinculin, talin, will attach to integrins, the same integrin. The only difference is now the integrin will be alpha 3 beta 1. If it's part of hemidesmosomes, it will be alpha 6 beta 4. Clear? So in focal adhesion, the integrins are integrin alpha 3 and beta 1, and they can attach to laminins 332 or 331. This is entirely a separate structure. This is not hemidesmosomes. These are known as focal adhesions. Now, focal adhesions are additional attachments apart from hemidesmosomes. They attach to actin filaments. This is very important, not keratin filaments, and they provide additional support. Okay, but if there's mutation in any of the proteins which are part of focal adhesions, then you can have blistering disorders like we see in Kindlin mutation causing Kindler syndrome. Okay, so let's go to Kindlin. Kindlin is a major inside-out integrin activator. Now, what do you mean by that? Inside-out is, we know from the previous diagram that Kindlin is inside the cell and they're going to activate integrins so that they can attach themselves to the lamina densa. So the signals to attach come from inside the cell to outside the cell. So they are the major inside-out integrin activators. So basal keratinocytes have Kindlin 1 or 2, which are formed in homologs 1 and 2. Vinculin, talin, these are additional molecules and they provide focal adhesions. Mutations in the firm T1 gene, which encodes for Kindlin 1, is the cause of Kindler syndrome. Kindler syndrome is an autosomal recessive syndrome which is characterized by trauma-induced blistering, photosensitivity, poikiloderma, variable mucosal involvement, and increased risk of mucocutaneous SCC. So remember that focal adhesions, they may not be the major part of attachment of basal cell to the underlying tissue, but the mutation in the protein can still cause trauma-induced blistering and photosensitivity and poikiloderma and can increase the risk of mucocutaneous SCC. So here is Kindler syndrome. It's a very good short note or very good question viva. Maybe in future we can discuss Kindler syndrome in a short video. Okay. So with that, we'll finish our discussion on the second part of basement membrane zone. This time we have covered hemidesmosomes and we have attached itself to lamina densa. In the third part, which will be the final part of basement membrane zone, we'll discuss the attachments of the molecules to lamina densa and the further attachment to anchoring fibrils. And then we'll finish our discussion on basement membrane zone. And if you go through all three of these videos, it will be very easy to understand. For me, it was tough during my residency to understand how a desmosome is formed, how a hemidesmosome is formed. It was so difficult to understand what all proteins are where. But now, if we know the location of these proteins, we will know what will happen if any autoantibodies are secreted against them or if there are any mutations against them. Okay, so if you know the location, you will know that mutation in, for example, integrins will cause subepidermal blistering disorder. Okay, while mutations in desmoglins are going to cause intraepidermal or suprabasal split. Okay, so now you know, because these proteins are present at a certain location, mutation or destruction of these proteins will lead to the split at that level. And mutation superficially will have EB simplex, and in between the cell and the dermis will have junctional EB. So some kind of concepts should be clearing up how the split happens. Okay, so without much further ado, I would like to just say thank you. Any suggestions, any comments, you can always put your queries in the comment section. It becomes easier for me to answer. One more suggestion that I would like to give is, if you are asking a particular doubt about a particular part of the lecture, it will be beneficial for me if you can mention the time of the time lecture. For example, let's say I say something at 36 minutes 2 seconds and you have a doubt regarding that. You mention this time in your comment so that I can see what exactly is the context of your query and then I can answer it that ways. Okay. So with that being said, we'll finish this week's lecture. Adios. Enjoy your weekend and bye bye.